Thank you, everyone. So our first speaker for today is Ann Kessler from NYU. And we'll give you a mark at like 4 minutes or so. And then if folks have any questions, if they can post them into the chat, and then we'll do a Q&A session afterwards. And if you just want to say next to go to the next slide and things.
Sure. Great. Well, thank you to the organizers and thank you to everyone for being here. It's great to be able to talk about how we are detecting Plasmodium infections in a cluster-assigned quasi-experimental study that's looking at the effectiveness of malaria camps in Odisha state in India. Next slide, please. So Odisha state has historically carried a large proportion of India's malaria prevalence. As you can see from the state map, much is in red, which indicates a high API greater than 10 and includes our study region, which is in the northern part of the state. Next, please. In response to this high malaria presence, the Odisha State Malaria Control Program started an initiative that they call Malaria Control in Inaccessible Areas. This includes an intervention called malaria camps that are set up multiple times per year in villages that are in remote and rural forested areas of the state that do have a lot of malaria. These malaria camps consist of mass screening by RDT of all villagers and treatment of positive cases. And it's accompanied by intensified vector control as well as community education and mobilization. Next, please. We are evaluating the effectiveness of these malaria camps at reducing malaria prevalence in a quasi-experimental study that has 3 arms, phase 1 of which happens over the course of 2 years. The first arm, Arm A, which we call New MC, consists of 6 villages that received the malaria camps for the first time in 2019, and again, subsequently in year 2 of the study, 2020. The second arm we call Delayed MC. This is a crossover. These are 6 villages that do not receive the intervention in year 1 of the study, but do receive it in year 2. And then we have a third control arm of 3 villages that we call Old MC. These are villages that have historically received malaria camps before the start of the study and continue through the 2 years of the study. You can see in the bottom line of the timeline in black, we have indicated our study visits, which occur in parallel to the administration of the malaria camp intervention, and we sample and survey all participants immediately before the intervention is received. Next slide, please. At each of these study visits, all study participants are evaluated for Plasmodium infection by microscopy, by bivalent RDT, as well as PCR, with our primary outcome being Plasmodium prevalence and species as detected by PCR. Next, please. The study participant workflow consists of consent, clinical questionnaire, anthropometric measures being taken, and a small volume of blood for the quantification of hemoglobin, of course, the RDT and the blood smear, as well as a small volume of blood for molecular assays. Next, please. The molecular assays are completed at our partner institution, Community Welfare Society Hospital in Rourkela, Odisha. And these assays consist of a species-specific PCR that identifies novel multi-copy targets for both falciparum and vivax infection. We use a Falsivax RDT. This is done in the field, and it identifies HRP2 for falciparum infections, which do largely predominate in the region, and PBLDH for vivax infections. Because of this predominance of falciparum and because we're using an HRP2 identifying RDT, we do conduct PF HRP2 deletion assays by PCR targeting exon 2 for any RDT negative PCR positive individuals. Next, please. As well as a number of serology and sequencing and entomology assays, which I will not discuss today. Next, please. So looking at our baseline survey data only from all arms, about 2,400 individuals, we found a Plasmodium infection rate of about 10% in this region. Of these 240 infections, 80% of them were submicroscopic. This means that they were not detected by RDT, they were only detected by PCR. Next, please. We have started evaluating these submicroscopic infections for PF-HRP2 deletion because we know that it is present in Odisha state. And from the 110 samples that we've evaluated for exon 2 so far, the large majority, 97% of them, appear to have an exon 2 deletion. And so this will continue preliminary data, but we're just getting started with it. Next, please. And looking across all survey rounds and study arms, just over 4,000 samples assayed, we evaluated the sensitivity and specificity of the Felsivax RDT against the gold standard of PCR in this study region and found a sensitivity of about 19.2%. Next, please. So in summary, the current testing methods, which is RDT only, that the malaria camps are using are likely missing the majority of Plasmodium cases in this study region, and that RDTs employing antigens other than HRP2 or the incorporation of molecular methods would greatly improve the effectiveness of the malaria camps mass screen and treat component. And that's all that I have.
Thank you so much for your presentation. I can get us started with questions, and if folks have other questions, please sort of post them in the chat. So I'm not really familiar with malaria in India, but is it common to have such low sensitivity for that RDT in India? Like, I mostly work in Sub-Saharan Africa, so it looks really bad to me. Does it also look bad to you?
Yeah. So I'll be honest, we were quite surprised by this low sensitivity. I will say Odisha state in particular in India, and India at large, has done a tremendous job in bringing down symptomatic malaria rates nationwide, but specifically in Odisha, they've done quite well in the last 5 to 10 years. So we are moving into a space where there's more asymptomatic and more submicroscopic infections. But yes, we too were a bit floored by this low rate. We do know, though, in this area of India that HRP2 deletions are relatively common. So.
Thanks. There was also a question with Tamaki about the parasite loads for the submicroscopic infections. Sort of what are they?
What are the parasite loads for the submicroscopic infections? You know, we haven't evaluated that, quantified it down to like a parasites per microliter, or I don't have the blood smear data either. I see the second question from this individual, if we've looked at HRP3 deletions. We're starting with the exon 2 deletion. We have started looking at the exon 1, exon 2 encompassing area and potentially doing some sequencing, but no, we have not looked at HRP3 deletions yet. And
yeah, do you want to answer the next one?
The next question, do you have any historical data showing growing numbers of parasites with HRP2 deletion? In this specific area, I'm not sure about growing numbers. But it has been documented by a number of groups, some in-country control groups as well as research groups, that there are a number of pockets in this part of India where it's relatively common, but I'm not sure if there's data showing that it's grown in the last years or decades.
Great, thank you so much for your presentation.
Thanks.
So the next speaker is Hannah Ehrlich from Yale University.
Great, thanks, Amy. All right, hi everyone, my name is Hannah Ehrlich. I am part of Sunil Parikh's lab at the Yale School of Public Health, and I'm going to jump right in here. Antimalarial drug resistance has arisen rapidly to monotherapies and spread historically along the directions shown by the arrows. The same is occurring regionally for artemisinin-based combination therapies, or ACTs, which is our current standard of care. An important tool that we have to track the emergence and spread of drug resistance is to look for point mutations in key loci associated with drug resistance, including 2 transporter genes, PFCRT and PFMDR1. For the most common ACTs used in Africa, specifically artemether-lumefantrine and artesunate amidiaquin, resistance appears to be selected for in opposing directions based on in vitro and field studies. So for example, the wild type PFCRT K76 is selected for after lumefantrine treatment, whereas the mutation 76T is selected for after amidiaquin treatment. Next slide, please. The good news is that researchers have been tracking these markers over the last 2 to 3 decades. So we asked, who is doing this work, how often, and how can we harness the existing data that we have? So we conducted a systematic search for studies that collected data on 4 of these markers related to ACT partner drug resistance, which are shown in bold and in orange, from 2004 to 2018, representing the uptake and widespread usage of ACTs on the continent. We compiled 254 studies together. And what we found was that many areas exhibited limited spatial data coverage. So just one example, in Côte d'Ivoire in the top right, the surveys, or the black dots, are clustered around cities, whereas the majority of the malaria burden occurs in the area shown in red. We also observed an average delay of over 3 years from sample collection to publication, and other concerns for the general sustainability of this mode of surveillance. And this work has recently been published in the Lancet Microbe. Next slide, please. We can use Bayesian spatial models to develop interpolated maps, which incorporate regression parameters and spatial autocorrelation parameters to estimate what is happening in regions between data points, for example, in most of Côte d'Ivoire. And we borrow from what we know about from data that we do have. This work is in preparation for submission, and here we can see that over time, moving from the first column of maps to the second, that the molecular landscape of drug resistance has changed dramatically over 15 years. We can see evidence of strong selection of the markers that confer resistance or reduce susceptibility to artemether-lumefantrine, AL. And for the first time at the scale for PFCRT, we can see evidence of a continent-wide resurgence of chloroquine susceptibility, confirming many local studies that have found the same. We also saw strong spatial dependency in the data, suggesting that fixation of some of these markers is due not only to changes in drug pressure and fitness costs, but also the spatial network of parasites themselves. So final slide. To make these maps, we incorporated, as I previously mentioned, multiple regression parameters, or parameters that we hypothesize to be predictors of drug resistance at the population level or ecological scale. What we found was that, for instance, areas with strongly seasonal malaria transmission tend to be correlated with a lower prevalence of mutations known to have fitness costs. And finally, in another application of these maps, we can use our results of our models to strengthen local and regional surveillance. If we subset our results by national first-line drug policy, we can identify regions with strong selection of markers over time, specifically the markers that are associated with resistance against that particular first-line drug regimen. And these areas are shown in red and orange, and we think that they may be considered of high priority for future surveillance efforts and local decision-making when altering partner drugs. That's all. Thank you.
Thanks so much. If folks have questions, please put them into the chat, but I can get us started. So how do your maps sort of compare with the different drug policies? Like some places will use certain drugs for pregnant women or not, or they do mass campaigns and things. So does it sort of line up where you think there should be a lot of issues with resistance or not?
Yeah, great question. So perhaps the biggest limitation of this work so far is that we don't have detailed data on drug consumption. And so right now what we're working with is looking only at first-line drug policies to treat confirmed Plasmodium falciparum malaria. And so we aren't considering things like malaria treatment in pregnancies or seasonal malaria chemoprevention in West Africa. However, that is in the works to see if that, and particularly SMC across West Africa, does change our results. However, that initiative is relatively new, and these models look at data collected from 2004 to 2018, but most of the data collection ends around 2015, which is right around the time that SMC administration began in West Africa. So we're not expecting to see major changes to our results there.
Great, thanks. We have a couple more questions. So one is, can you describe what the uncertainty is in slide 3?
Yeah, thanks for that question. I should just specify it on the page itself. So that is the standard deviation from the posterior prevalence estimates that we get out of our MCMC results from our models. So it's standard deviation.
Great, thanks. Tamaki had a question about why you think seasonality might be so important.
Yeah, that is a great question. We were surprised to see that it consistently was the strongest predictor for almost every marker in terms of the prevalence of drug resistance in an area. We think that it might be sort of protective against mutations with fitness costs. So particularly, so in regions with high seasonality, there's a long period of time in the dry season where there's limited malaria transmission and therefore limited drug use or drug pressure. And so we expect that in those regions, there's some sort of reversion to the wild type because of fitness costs being associated with the mutation, if that makes sense.
Yeah. And then there's another question that's sort of about seasonality in general and about how MDA might be related to seasonality or change or interact and what other climate factors and how that would play out in terms of resistance.
Yeah, so we incorporated a number of climate factors related to, a number of climate parameters in our models themselves. And we did, just to start off, for example, we really only saw seasonality as a strong predictor with rainfall, but the two were relatively collinear. In terms of MDA, I think that a helpful analysis moving forward would be to consider regions that use MDA, which I believe tend to be regions with more seasonal transmission. And so I think that those two issues would be correlated in terms of their respective results or impact on drug resistance.
Great, thank you so much. So now we're going to try again to have Jonathan Manyami present, who's from the University of Health and Allied Sciences in Ghana.
Okay, thank you for the opportunity. I am Jonathan from the University of Health and Allied Sciences, Ghana. I would like to thank my research supervisors for the good work done in producing this work. I present the study on effects of pre-consultation testing on clinicians' adherence to malaria test results in the northern zone of Volta region, Ghana. Next slide. In Ghana, the Ministry of Health has recommended that malaria treatment should be based on test results, which is to mean when the test is positive, that is when the malaria anti-malaria drug should be issued. However, despite the guidelines that has been given, research has found out that several patients are given anti-malaria drugs without confirmation. And there are some clinicians that do prescribe anti-malaria drugs to patients even when the test is negative. As a result, we conducted this study to determine if testing all children aged 6 to 59 months with fever for malaria at the OPD before consultation with a clinician, which we term pre-consultation testing. We're looking at if this system of testing the children before they went to see the clinician will influence the clinicians to adhere to the malaria test results. Next slide. In order to conduct this study, we used a quasi-experimental study design. This study design involved two studies, two health facilities. In one of the health facility, which was the intervention study arm, and the other was the comparative study arm. In each study arm, we recruited 439 children that were under 5. In the intervention study arm, all the eligible children were tested with the malaria rapid diagnostic testing before they consulted with the clinician, which was the pre-consultation testing. However, in the comparative arm, the services which involved the patients seeing the clinician before the clinician decides whether to send the patient for testing or not, we maintained that system which is already in place to compare with the system that we employed at the intervention arm. Next slide. The results of the study. Of the 878 children that were screened, I would like to start with the malaria prevalence. The overall prevalence of malaria was 31.9% by the RDT, and 26.7 by microscopy. We realized that the clinicians in the intervention arm adhered more to the malaria test results as compared to those in the comparator arm. Also, the proportion of children who tested negative but were still diagnosed by the clinicians with malaria was significantly lower in the intervention arm as we compared it with the comparator arm. Also, the time spent, because that was one of our study objectives. With regards to the time spent, on the average, a patient's waiting time was equally significantly lower in the intervention arm, which was averagely 2.61 hours, as compared to the comparator arm, which was on the average 3.42 hours. So based on our study results, the following conclusions were made. We concluded that pre-consultation testing significantly improves clinicians' adherence to malaria test results and shortens patients' waiting time at the OPD. Based on our conclusion, we recommend that the National Malaria Control Program should consider establishing RDT canals at the OPD of the health facilities to implement pre-consultation testing, which is the children should be tested for malaria with the RDT before consultation with the clinicians. Thank you so much for your hearing.
Thank you so much for your talk. So if anyone has any questions, please post them into the chat, but I can get us started. So in your study and in your experience, did you find that the clinicians liked this process better? I think you're able to show what the effectiveness might have been, but did you do any interviews with the clinicians and ask them how they liked the different systems?
Okay, yes, we did. We did interview the clinicians as well. And on that, we asked them which of the two systems did they prefer. And on the average, most of the clinicians preferred the pre-consultation testing, owing to the fact that it made it easier because the children come with the test results, which makes it easier for them to prescribe their drugs accordingly. So yes, we did interview the clinicians to look for their preference. Thank you.
Yeah, and on that question, so there was a follow-up question about, are antimalarial drugs available only through the doctor's consultation, or could people get them at the pharmacy?
Okay, yes, there are those who get them at the pharmacy, but the policy indicates that for one to get an anti-malaria drug, it should be as a result of a testing. But we do know that some of the pharmacy shops have the RDT within the facility. So if someone goes with the symptoms, they do test them for the malaria before issuing the drugs, because some pharmacy shops do have clinicians in there. But the policy states that no anti-malaria drug should be issued unless the test says it is positive. Thank you.
Great. And there is one final clarifying question about what statistical test did you use for significance?
Okay, with the significance, because we didn't look at the factors that are associated with the adherence, we didn't actually look at it, but we used stata, and significance was looked at 0.05, and we used the chi-square to look at the strength of the association. Thank you.
Great. And there's one final, question, since we have some time, about, did the doctors miss other diagnoses due to the positive malaria test, or is it not very common for children to present with non-malaria fevers?
Okay, based on our study objectives, yes, when the children tested positive for malaria, there were, it made it easier for the clinicians to issue the anti-malaria drugs. But in the case where the test was negative, but the doctor still issued the anti-malaria drug. So we recommend for a further study why they will do that. But as to what was done with the other conditions, that was not our study objective. Therefore, we didn't investigate into that. But we sure will do that in the future. Thank you.
Great, thank you so much. And thank you, everyone. That's all the speakers that we have for this session. So on behalf of the organizers, we want to thank all of you for attending the Future of Malaria Research Symposium. This event was sponsored by the Johns Hopkins Malaria Research Institute and would not have been possible without the leadership and support of Dr. Peter Agre and Genevieve Williams. We're also grateful for the technical support from Sean Rogers, Tyler Hill, and Cone, without who this day would not have been possible. 2020 has been a tough year, but one of the things that we hope is one of the couple silver linings of this year is that the ability that we're able to hold this symposium where so many more people are able to attend from all across the globe, and we're able to have a much more diverse range of speakers presenting on a lot of different topics in malaria. So we're really happy and grateful for everyone who's attended, for everyone who came to share their research, and all the speakers who participated today. And we hope that you'll all join us next year for World Malaria Day, which will be on April 23rd, 2021. Thanks everyone for attending, and continue about your Zoom-based lives. Thanks everyone.
Thank you.